Thyroid gland is a butterfly-shaped organ. It is located anterior to the trachea, just inferior to the larynx. It is divided by a isthmus, which is a medial region that can divide it into a left and right lobes. Each thyroid lobes are embedded with the parathyroid glands. These parathyroid glands are 4 in number. The tissue of the thyroid gland is composed of thyroid follicles. Inside the follicles, the colloid is present, which is a sticky fluid, and inside colloid, the glycoprotein is present, which help in the formation of thyroid hormone. And the production is dependent on the hormone essential, and the unique component is the TS of thyroid gland, in which the colloid containing follicles, and outside it the parafollicular cells are present, follicle cells, which is made up of cuboidal epithelium. Hormones are produced in the colloid. When atoms of a mineral iodine attach to a glycoprotein, then it is called thyroglobulin. This thyroglobulin is secreted into the colloid by a follicular cells. The following steps outline the hormones assembly. First, the binding of TSH, that is thyroid stimulating hormone, to its receptor in the follicle cells of thyroid gland, that can cause actively transport of iodide ion across their cell membrane from the bloodstream into the cytosol, by which the concentration of iodide ion trapped in the follicular cell is higher than the concentration in the blood. Inside the follicle cells, the ion undergo oxidation. So the oxidation of two iodide ions result in iodine, which passes through the follicle cell membrane into the colloid. Third, in the colloid, the peroxidase enzymes are present. This enzyme helps in linking of iodine to the tyrosine amino acid in thyroglobulin to produce two intermediates, a tyrosine attached to one iodine and a tyrosine attached to two iodines. When one of each of these intermediate is linked by a covalent bond, the resulting compound that is formed called triiodothyronine, that is T3, a thyroid hormone with 3 iodine. Much more commonly is the two copies of second intermediary bond forming tetraiodothyronine, also known as T4, a thyroid hormone with 4 iodines. This is the regulation of thyroid hormone level. When T3 and T4 concentration in blood is low, the hypothalamus releases TRH at that time. This triggers the release of TSH, that is thyroid stimulating hormone, by anterior pituitary. The TSH release help in increasing basal metabolic rate and rise in body temperature. The elevated T3 and T4 level inhibit the release of TRH and TSH, whereas the high T3 and T4 level stops the TRH release, and the anterior pituitary also stops the release of TSH. These are the two aspects in which we have seen that if the concentration of T3 and T4 is low, and if the concentration of T3 and T4 is high, in both cases the pituitary gland either inhibit or stimulate the TRH hormone. That can show a classic negative feedback loop control.
She got